We're on the traditional lands referred to as Treaty 4 territory, the original lands of the Cree, Ojibwe, Saulteaux, Dakota, Nakota, Lakota, and on the homeland of the Métis Nation. We respect and honor the treaties that were made on all territories, and we acknowledge the harms and mistakes of the past. We are committed to moving forward in partnership with Indigenous nations in the spirit of reconciliation and collaboration. Dr. Ryan Fisher is the Curator of Vertebrate Zoology here at the Royal Saskatchewan Museum, and we are fortunate to have him speak with us this evening. Now, if you check out his page on the museum's website, there's a lot of excellent information about Ryan. He's done a lot of research. His work is so much more than just dead birds. The Royal Saskatchewan Museum's website. So his publications are going back to 2004, for example. They include prioritizing recovery funding to maximize conservation of endangered species, northern pike, ground squirrels, conservation strategies, ferruginous hawks, responses of songbirds to climate change, burrowing owls, Sprague's pipit songs. I could go on and on. So I'll turn this over to our speaker and welcome to Dr. Fisher.
Well, thanks very much, Fran. I hope everyone can hear me okay. Hopefully the audio is coming through strong and you can hear me all right. I'm just presenting from home tonight, but hopefully it'll give you a little bit of an idea of what I do here at the Royal Saskatchewan Museum, sort of on a day-to-day basis. So I'm just going to turn my camera off and turn my screen sharing on for the presentation. And cross my fingers that everything works. Hopefully everyone can see that okay. So, I hope everyone can see this okay. Like Fran said, I am the Curator of Vertebrate Zoology here at the Royal Saskatchewan Museum. So I always like to start a presentation telling you a little bit about myself. There's lots of familiar names in the participant list, and I'm sure there's lots of familiar faces in the live audience tonight. But for those of you who I don't know very well, I'm a Regina kid, a prairie kid. I grew up in Regina. I had done my master's degree at the University of Saskatchewan on northern flickers, so woodpeckers. And then I completed my PhD at the University of Regina on grassland songbirds. Over the last about 7 years and a little bit, I had the good opportunity to work with both federal and provincial government wildlife agencies. And you know how we always talk about, people who have bought a new dog and they have a pandemic puppy. Well, I'm a pandemic curator, so I'm fairly new to this position. I just started at the Royal Saskatchewan Museum in December of 2020. And I'm also an adjunct professor at the University of Regina. So this just means that I can co-supervise students and teach classes at the U of R, as well as my sort of day-to-day duties at the RSM. So I think Fran covered it pretty clearly about what I've been up to sort of in my wildlife career. So I had the good fortune of starting my work in the wildlife realm, working actually for the former curator of vertebrate zoology, Dr. Ray Poulin, on burrowing owls, way back in the early 2000s. Around that same time, I also had the good opportunity of working with Dr. Mark Brigham on common nighthawks in Cypress Hills, Saskatchewan. A few years later, I came back to the University of Regina to work on my PhD, focusing on grassland songbirds, but with a real emphasis on Sprague's pipit. And so I did a lot of my field work in the Last Mountain Lake National Wildlife Area, the north end of Last Mountain Lake. More recently, for some postdoctoral fellowship work, I got another opportunity to add an iconic prairie species to my list and had the opportunity to work on ferruginous hawks, and kind of went back to my roots a little bit and did some work on burrowing owls again. And then in the last couple years, I had the good fortune of co-supervising a student, Brandon Berta, who did some research on our provincial bird, the sharp-tailed grouse, here in Saskatchewan. So all of my sort of research experience obviously has been focused on birds, as you can tell from the pictures here, but also with a focus on our prairie ecosystems here in Alberta, Saskatchewan, and Manitoba. Now, a lot of the stuff that I'm going to talk about tonight, you'll see the fingerprints of our curatorial assistant, Danae. A lot of the stuff that I'm going to talk about our collections management, preparation of specimens, and kind of everything else under the sun is really driven by Danae at the RSM. So Danae did her bachelor's degree and her master's degree at the University of Regina. And just lucky enough, Danae had done her master's degree on bees, and we managed to convert her to a bird person, or at least we think so. And if people don't realize, there is a little bit of a friendly rivalry between the invertebrate and the vertebrate zoology parts of the RSM. So like I said, a lot of the stuff that I'm going to talk about tonight, you'll see a lot of the fingerprints of Danae's awesome work at the RSM. So the Royal Saskatchewan Museum has kind of three main mandates, and if we sort of think of that from a vertebrate zoology standpoint, our first mandate is to collect, study, and preserve specimens and information about Saskatchewan's natural history. We also want to interpret and communicate that knowledge to the general public. And at the RSM, we also actively do scientific research on Saskatchewan's natural history and wildlife. So my talk tonight is going to sort of focus on these three parts of my job here at the RSM, and I'll specifically be sort of focusing on the first and the last bullet there. So most everyone is probably, I hope, familiar with the main Royal Saskatchewan Museum building. This is where all of our amazing exhibits and dioramas are located, and where a few of you are sitting live in the auditorium tonight. So you can wander through the RSM and see some amazing creatures and some amazing displays ranging from nesting golden eagles to moose, to some amazing northern dioramas of woodland caribou. And this is probably where most people have their interactions with museum staff and with the museum's collection. But how I like to think of it is that the real magic happens elsewhere, or behind the scenes. And that happens at 2340 Albert Street, which is just kitty corner from the main museum, and right next to Leopold's Tavern, if anyone needs a little bit of a landmark. So let's jump right into our collection at the Royal Saskatchewan Museum of specimens related to Saskatchewan's natural history. So the RSM collection, vertebrate zoology is just one collection and what I'll be kind of focusing on tonight. But we do also have an invertebrate zoology collection, which is curated by Corey Sheffield. We have a botany collection, which is also curated by Dr. Sheffield. We have a paleontology collection, which is curated by Dr. Ryan McKellar. We also have an Indigenous Studies collection, which we are currently looking for an Indigenous Studies curator to hire at the moment. And we have two other collections, the library media and a fine art collection. So the museum has sort of 7 main collections. And like I said, I'm really going to be focusing just on the vertebrate zoology collection tonight. So put on your socks and let's have a quick look at some of the interesting things and specimens and how we store them over at 2340 Albert Street, where the real magic happens. So our primary collection is made up of usually specimens that come from the wild in Saskatchewan. And it's really intended to represent vertebrate biological diversity over space within Saskatchewan and also over time. And to make that collection relevant and usable from a wide variety of potential users. So when I first started at the museum, I was kind of curious where all of the specimens had come from at the RSM, at least as it relates to birds. So the map on the right, all those little dots show where we have birds that were collected in Saskatchewan, in North America, and even overseas. So it's pretty interesting that we can open a drawer in our collection and potentially find something from Greenland sitting in there. So at the end of 2020, we had about 9,300 bird specimens represented by about 421 species. The picture on the right is typically how the birds are stored, and I'll talk about that a little bit later. But this is one of my favorite collections just because of the amazing color that we can see in a lot of the wood warblers and tanagers and a few other species here in Saskatchewan. Our mammal collection is just over 3,700 individual mammals represented by 126 species. We have 519 individual reptiles, 125 amphibians, both of those represented by about 105 species total. And we have just over 350 fish specimens represented by 64 species. So we got birds, mammals, reptiles, amphibian, fish sort of individuals in our collection. One of the other neat aspects of our collection here at the RSM is our collection of owl pellets. And to my knowledge, it's one of the only collections of owl pellets in the world. So the RSM stores about 10,000 owl pellets, and some industrious graduate students, curators, and curatorial assistants went through those 10,000 owl pellets and identified the remains of about 92,000 vertebrates within those owl pellets, representing birds, mammals, reptiles, and amphibians. So for those of you who don't know, when owls consume something, all of the little bits that they can't digest, they regurgitate or they throw up in a pellet. And you can take that pellet, you can dissolve all the nasty hair and leftover soft tissue parts, and you end up with a bunch of bones. And through some really good detective work, you can tell what type of mice or what type of birds that owl was eating. So as you can see, it's a really, really unique collection that we have here at the RSM, and was one of the big projects that Dr. Ray Poulin led when he was the curator of vertebrate zoology. Now, we also have a nest and egg collection at the RSM. So our egg collection consists of about 789 different individual eggs, ranging from hummingbird eggs, clay-colored sparrow eggs, all the way up to Canada geese and some of the bigger species. So quite an amazing diversity of eggs. I tried to take pictures of possibly the most bland eggs that are brown and white as possible, but those are a lot of the species that we have here in Saskatchewan that rely on some type of camouflage for their eggs. Our nest collection consists of about 260 nests. We have everything from big raven stick nests to floating nests from grebes to smaller clay-colored sparrow and hummingbird and oriole nests. So a really, really interesting collection and all pretty much local to Saskatchewan breeding birds, which is pretty cool. So you might be asking yourself, well, where do all of these things that end up in the museum come from? Well, historically, a lot of specimens were actively collected for the sole purpose of adding them to the museum collection. So it wasn't uncommon for people to go out and shoot a bird to add it to the collection, or collect a nest, or collect eggs to add specifically to the museum collection. Now, these days, we don't do much, if any, active vertebrate collection other than maybe for owl pellets. And our collection mostly increases through donations from wildlife rehabilitation organizations. So we have a great relationship with Living Sky Rehabilitation and Salthaven West here in Regina. If you ever drop anything off to conservation officers, a lot of that, those birds or mammals end up in the RSM collection. We have a really, really great relationship, just fairly new over the last couple of years, working with Nature Regina on their Bird Safe Initiative program. So any birds that folks pick up as part of that volunteer program are donated to the museum. And we get some pretty amazing birds coming in as part of that program. This year it was neat to see, and somewhat disheartening, but it was interesting to see a rusty blackbird come in, quite a few clay-colored sparrows, and just stuff that we might not think of as migrating through Regina. So real big props to Jeff Gamble and Nature Regina for donating all the stuff you find to the RSM afterwards. We also do get a lot of submissions from the general public. So if folks find a dead animal in their backyard, they can donate it to the museum, and I'll talk a little bit about that a little later. So how are specimens prepared and stored once we kind of get them at the museum? So the vast majority of our collection, at least of birds and mammals as well, is in something called a study skin. Now, this isn't a lifelike pose, as you can see in the slide. I tend to call it bird on a stick, but study skins do allow us to store a large amount of, or a large number of specimens in a relatively small space. So you can see all those wood warblers in the cabinet. But it also allows us to maintain useful scientific information. So we still have a lot of information remaining on the coloration of plumage or fur. We can also get a lot of good information still on things like the shape and size of different specimens that come in. And it also preserves a lot of information on fur, feathers, skin, etc. So like I said, the vast majority of things that we have over at the annex in our collection are in study skin pose. Now, sometimes stuff comes into the museum that's maybe in a little bit of rough shape, and the feathers or the fur just doesn't lend itself well to making a study skin. But in those cases, skeletons tend to be almost as valuable to us, or it sort of makes the best of a bad situation. So skeletons can provide us information on the size and the shape of the animal and its different bones. It can also give us potential information on injuries that the bird or the mammal or the reptile or amphibian may have suffered before being donated to the RSM. And we can really make skeletons out of anything. So the top picture shows a small skeleton of a robin that was probably submitted to us as part of a window strike, to very, very large skeletons like the wolf that's in the bottom picture there. So we make, and when I say we, it's mostly Danae, and we sometimes have some summer students working on this. But we can create skeletons from all sizes of things that come into the museum. Now, the other way that we store things at the museum or have specimens of is taxidermy mounts. Now, these are, everyone's probably familiar with them if you've gone to the main exhibits or if you've done some shopping in Cabela's. These taxidermy mounts exhibit lifelike poses and behaviors, but taxidermy mounts really encompass a significant investment in time and money to prepare. They also take up a lot of space because they are in those natural poses. So, you can imagine what a golden eagle in a lifelike pose, how much space that takes up in our collection. But these taxidermy mounts do provide useful information on things like coloration, shape, and size, still preserves the fur, feathers, skin, etc. So here, currently at the RSM, we actually don't do any in-house taxidermy, and all of our preparation methods are focused on study skins and skeletons. The taxidermy mounts, we continue to get some donations of taxidermy, and a lot of them tend to be older taxidermy that was done in-house or out of house and then donated to the museum. So I like to call these ones the pickled bunch. We do have a significant number of things that are stored in liquids at the RSM, and it's mostly our fish, reptile, and amphibian collection. Things that are easy to plunk into a jar and preserve with some type of preservative. They're extremely long lasting. But at least for us, storage is difficult just because of the large quantities of potentially flammable material or liquids that are being stored at the annex. But it is interesting to go through some of the jars and see things like bull snakes and rattlesnakes, some different types of fish, Western painted turtles like you might find just out in Wascana here. So we also store tissues of each specimen that we prepare and add into our collection. So we store samples of the liver, the heart, and the muscle for each thing that enters into our collection. Those are stored in perpetuity in a -80 freezer. So prior to the pandemic, you might have wondered if this was a Jeopardy question, what were -80 freezers used for before storing vaccines? Well, one of the reasons for needing that type of freezer is to store biological specimens. And we have one of those freezers at the RSM where our tissue samples are stored. Tissue samples are a really, really interesting thing because you never quite know what use people might have of them in the future. I'm sure if you ask curators 10 or 20 years ago what they could use tissues for, I'm sure that's completely changed in the last decade or so. So can use tissues for anything from genetic analyses to analyses that can tell you what type of diet the animal was eating prior to it making its way to the RSM. So we really don't know what potential is out there, but it seems to be ever expanding what researchers can use tissues for. So what happens if you drop a bird off to the RSM? So that bird is then taken in by our staff. Myself or Danae will usually go through an identification check just to make sure we've got the right species identified in whatever you brought in. Each of those things is given a unique barcode. And then it's put into our walk-in freezer to await preparation. Now, once we've selected something that we want to prepare as a study skin or a skeleton, the first thing that we do after it's unthawed is we record a ton of data and a ton of information on that particular specimen. So we'll note whether it's male or female. We take a lot of measurements. For example, for this flicker, we'll measure the length of its bill. We'll measure what its wingspan is, how long each wing is, the length of its tail. we'll take some notes about its plumage, different colorations, anything unusual about the bird. And then once we start doing the preparation and we are able to look at the insides of the bird, we can also get a little bit better understanding of whether or not it was breeding or it had bred in the past. For some birds, it's tough to tell males and females until you actually get inside of it. So we record all of that information. We'll even record what its fat levels are like. So for birds, you can imagine that certain times of year, they're putting on a lot of fat. And then at other times of year, they might be pretty skinny. So we really try to record as much information as we can before we do some preparation on it. We obviously kind of, for lack of a better word, have to take the bird apart to do some of that prep. We have a fairly large lab over here at the annex where we do all of the preparation on our vertebrate specimens. So it's kind of, it's a surprise every day of what you might see back in this area. It could range from someone preparing a northern flicker to someone with a full-sized pelican on the table, even sometimes a cougar or a wolf skin hanging from the fume hood. So it's definitely not a place for the squeamish to work, but you do get to see some really interesting things as they come through the museum. Now, I am going to talk a little bit about how we make skeletons. So if you are a little bit squeamish, I would say maybe close your eyes for the next slide. So I'll give people a couple seconds to decide if they want to keep their eyes open or if you want to close your eyes. But one of the things at the museum when we make a skeleton is obviously we have to take off all the feathers. We have to take off as much muscle and meat of the bird that we can. And so, you know, in that respect, like I mentioned, this is what you'll see Danae doing a lot at the museum and some summer students as well. So if you are really interested in dissections and want to work at the RSM, give us a call. So we take all of the muscle and the tissue as much as we can off the bird. And then we actually have a colony of dermestid beetles, or sometimes called flesh-eating beetles, that we'll put the birds in and let the beetles kind of do the rest of the job for us. They'll take that skeleton with some meat on it, completely clean the bones until you have a nice shiny white skeleton at the end. So kind of a nice way for us to have some very eager helpers who are only paid by maybe breakfast, lunch, and dinner. All right, for those of you who closed your eyes, you can open them again. So once we have something prepared as a study skin or a skeleton, it's time for us to add it to the collection. So each specimen gets a unique number and a tag describing some basic information on it. So for the flicker at the bottom there, you can see we've identified that it's a northern flicker. It was collected in Regina, Saskatchewan in 2015. And then all of the information that we collected on it is entered in our electronic museum database so that we have a complete record of it in perpetuity and for as long as the museum is around. And then each of those specimens is added to a specific drawer or cabinet at the annex. So all of those cabinets along the left side of this picture are all filled with different things from around Saskatchewan that have been added to our collection. So I just wanted to share with you some really interesting highlights. Like I said, I'm only a few months into the job here, and it was quite interesting to just be able to walk around the annex and kind of sift through some of our electronic records to see what's in the collection. And these are some of my favorites. So our oldest specimen is a female wood duck from 1899. It's actually not a specimen from Saskatchewan. It was donated from Ontario. But this female wood duck is still looking pretty good, 122 years old, which is pretty amazing, and shows how well our collection is maintained over time. I think that's a really great testament to all of the previous curators and all of the current curators and conservators to make sure that our collection lasts as long as possible. Our oldest Saskatchewan specimen is a chestnut-collared longspur from 1905. Again, still looking pretty darn good for being over 100 years old. Our oldest egg in the collection was a common loon egg collected in 1905 as part, I think, if I've read my history correctly, as part of a British expedition in northern Saskatchewan around Sandy Bay. So again, we think of eggs as being extremely fragile, but our egg collection has some eggs that are well over 100 years old and in pretty darn good shape. One of my favorites is a taxidermy mount of a passenger pigeon. So for those of you that don't know, no one has seen a live passenger pigeon in my lifetime and probably my parents' lifetime, as they've been extinct since the early 1990s. But through, again, like I said, the excellent curation of our collection, I can have a look at a passenger pigeon that has been stuffed and preserved at the Royal Saskatchewan Museum. It's pretty neat to see. So there's some other interesting specimens too. Just as I mentioned, I'm a bird lover, and so it's neat to highlight some of the unique things in our collection, at least for Saskatchewan. So we have actually a pretty amazing collection of greater prairie chickens, which are extirpated here in Saskatchewan. That just means they were here at one time, but we don't find them here anymore. Greater prairie chickens being quite different from other grouse species like sharp-tailed grouse and sage grouse, but some people call grouse in general prairie chickens, but this is a different species. We actually have a fairly large collection of greater sage grouse. So this is a male looking pretty dapper in one of those lifelike poses I talked about earlier. He's probably ready to dance on a lek in the spring. We have this really cool taxidermy mount of a wolverine. Pretty amazing when you get right up close to it and see those claws. You definitely realize that it's something that you definitely don't want to tangle with. And something that's still on my life list that seems to be popping up all over on SAS Burgers lately, but I haven't had a chance to see one yet, but a red-headed woodpecker. And we actually have quite a few taxidermy mounts of red-headed woodpeckers, which is always neat to see something that you don't see too much out in the wild, but you're able to see right up and close in the museum's collection. We do have some very beautiful taxidermy. Some of these ones you think that we should have on display, but there just isn't enough room. But this pair of bear cubs is kind of my favorite taxidermy mount at the museum. And then there's also this amazingly colored red fox at the back there, sometimes called the silver fox up in northern Saskatchewan. So kind of interesting just to see a lot of the amazing human work that was done on some of these taxidermy mounts at the RSM. Now, I kind of thought it'd be neat to look up some notable Nature Regina members and their donations to the RSM. So I'm not sure, I saw one familiar name in the participant list, but interestingly, Lauren Scott, this was one of his first ones from our database that I could tell anyways, donated a yellow rail back in 1974. We found this gray-crowned rosy finch from a W. Pepper in 1972. I'm guessing that's Wayne Pepper, but I'm not 100% sure. There might have been another W. Pepper out there. Gary Seib had donated an indigo bunting in 2005. So birds, one of the other ways that we do preserve birds is we do take wing and tail and feather mounts of those species. It saves a little bit of space and allows us to prepare stuff a little quickly. But I believe this indigo bunting was actually from Regina, if I'm correct. And then I did see Paula Gertis on the participant list, and this is an interesting one from Dale, a peregrine falcon found in 1988. I believe it was found out at Condi. So it is kind of interesting as a bit of a history buff that we can go back in our database and see who were the main collectors of things in the past and in the present. So if you ever do want to donate stuff, your name kind of goes down in history and stays a part of the RSM, which is a pretty cool thing. So you might ask yourself, well, what is this collection used for? Well, like I mentioned, so our mandate is to document biological diversity over space and time in Saskatchewan to really understand how biodiversity is changing. So, I'm sure if you asked curators at the RSM 50 or 60 years ago, whether or not they had any fox squirrels from Saskatchewan in the collection, I'm sure the answer would be no. But nowadays, it's a fairly common item that we get donated to the museum. So there's things like that we can use the collection to track changes in Saskatchewan biodiversity. Because our collection is open, we do get a lot of requests from researchers. Just as a really neat example, we've been pulling up a lot of interesting historical information for Dr. Spencer Seeley from the University of Manitoba, who's doing a fairly large analysis on natural history biologists and brown-headed cowbirds across the prairie provinces. So we've been working a lot with Spencer to look up who's donated different samples, provide them some information on cowbird eggs in our collection, etc. So it's always kind of neat. You never know what an email request from researchers are going to ask you to do. Something that I didn't realize when I first started at the RSM here is that artists also frequently make use of our collection. So as everyone can imagine, sometimes a picture just doesn't do something justice, and people want to see something up close. So the picture on the bottom is a wood carver from Saskatoon that requested to see a great gray owl. And we were able to accommodate that request, and he's working on this amazing carving for a competition, I believe. Similar to artists, our collection is also used for educational purposes. Pretty amazing when kids are able to see one of those taxidermy mounts up close and personal. It's really a lot different than seeing a bird on a screen or a picture, or seeing it in the wild when it's 100 meters away and you're looking through your binoculars. So a lot of different uses for our collection that you might not have thought of. So the big question that we get asked a lot of is, what should I do if I find a dead animal? And for the most part, the RSM will happily take any type of dead animal you find, although it should be in reasonable shape. And we do sometimes have difficulties with big stuff that comes in. But as long as we can identify it and it's not sort of a smushed pile of feathers, we'll happily take it from you. There are some steps to follow, though, of course, that we suggest people do if they want to donate an animal to the RSM. So use gloves when handling anything that has died. If you suspect that it has died of a disease, please don't handle it, or handle it as little as possible. When you handle it, you can place the animal in a container. We did a lot of things in plastic garbage bags and Ziploc bags, which work really, really well. What's really important, though, is to write the date and the location that you found the animal on the container that you're going to submit the specimen to. So when we're dealing with a collection that's used for scientific purposes, it's really, really important that we know when and where the thing was found. Without that information, the specimen definitely becomes a lot less valuable for us. Now, we don't have to have, for example, an exact GPS location if you're in Regina. Simply saying the city of Regina is absolutely fine. But if you're roaming around out in the country and you can provide us with a GPS location or the nearest town, that's fantastic. So once you've got the animal in a container and you've written the date and the location on it, you can drop it off at the front desk and we will happily take it in. Now, I'm not sure the front desk happily is the exact word that we would use, but they will definitely take it from you and then pass it over to Danae and I for processing into the collection. And you can drop that off anytime during the RSM opening hours throughout the week. So the other thing that we do is, of course, we interpret and communicate that knowledge that we have and that we've kind of accumulated as part of our collection to the general public. And so curators do a lot of public education and outreach. So we give talks such as this. We do have an interesting posts every second week, although we've been a little bit missed a few in September, but we have posts called Bird Mondays that will appear on Facebook and YouTube. They're normally videos, but they can also include Twitter and Instagram posts. So we've done some interesting ones on bird identification, on citizen science, and a couple of really, really neat ones on the links between birds and Indigenous culture. So have a look at that if you are able. All you have to do is search Royal Saskatchewan Museum on YouTube or Facebook, and all of the videos are there. So curators also teach and guest lecture at university classes. We participate in volunteer activities. So I've had the good fortune of leading some Nature Regina bird walks on Migratory Bird Day with Shannon Chernick. So that's always fun to get out and do some field work. And over at the main building, we also help to develop some of the exhibits when there's a new exhibit on the go. Now, also, one of the important things about the collection is that that data should be accessible publicly. And so if anyone wants to know where specimens have been found in Saskatchewan and are held at the RSM, all of our data is publicly available on something called, or on a website called the Global Biodiversity Information Facility, or gbif.org for short. And on that website, you can, for example, look at the locations of where all the bird specimens came from, all of the fish specimens, all of the mammalian specimens, and download all that data for research purposes. As I mentioned, people can also directly reach out if they want more specific information on certain things in the collection. We field lots of different requests from researchers and from the general public about the RSM's collection, and we're generally happy to respond to those and help out where we can. Now, the other thing that we do at the RSM is all the curators are engaged in some type of research in the study of Saskatchewan's natural history and Indigenous issues. So let's talk a little bit about some of the research programs that I have going on at the RSM. So as I mentioned, curators typically work closely with University of the Regina researchers, although a lot of us lead or involved in various research projects outside of the University of Regina, but within Canada. And like I said, I'm fairly new here, but just getting 2 research programs off the ground shortly. And I'll talk about each one of these very quickly in the last little bit of my talk here. So I'm not going to talk too much about this one, but we're starting up a project on great horned owl habitat use and hunting behavior in southwestern Saskatchewan. So we're going to be attaching satellite transmitters to great horned owls to kind of study their movements and behavior. We're also going to be putting nest cameras at nests to understand the diet of owls a little bit better. And this project is going to be led by 2 brand new graduate students at the University of Regina, Tori or Victoria Hartley Cox and Dominic Smarta. One of the big parts of this project that we're hoping as well is going to be a little bit of a citizen science component to have people report great horned owl nests to us so that we can do some work on great horned owl nest site selection. So I'm not going to talk too much about this project because I hope that Victoria and Dominic can come back at a later time and tell you a little bit more about your research. But stay tuned. We'll have some exciting developments about this project in the future. So the other project that I wanted to talk about that is probably of very big interest to the Nature Regina group here is an urban wildlife project that I'm starting up. So myself and I'm working with the Batman, Dr. Mark Brigham at the University of Regina, and another provincial biologist, Dr. Leanne Heisler, on this research. So just to give folks a little bit of background on urban wildlife. So, we all kind of think of cities as a bit of a biodiversity desert, but that's not necessarily true. So there are natural and semi-natural areas in cities that provide important habitat for wildlife. And these same areas can and are also important for human well-being. There's some really interesting new research linking people's happiness levels and their ability to get outside and hear birds singing and experience nature. But when we're in a city too, most human interactions with wildlife are neutral. So, we may be driving down the road and we see a rabbit and we don't really think twice about it, or we see a robin, hopping around in the grass and don't think too much of it. We may have those really positive interactions with wildlife in the city. During the pandemic, being outside and bird watching was probably really, really good for everyone's mental health. But we do have to be honest and say that some interactions with wildlife in sort of an urban setting can be negative. And that's where urban wildlife research sort of fits in. So again, this is somewhat depressing, but cities are actually one of the newest and the fastest growing ecosystems on the planet. And while that's not maybe necessarily a good thing, it does provide a unique opportunity for science and understanding how wildlife is adapting to these new ecosystems. And what people have generally found is that as urbanization increases, biodiversity increases, or as you kind of go from the edge of a city, you probably find a lot more species than you do if you're right smack dab in the urban jungle. We know some species like raccoons have done really, really well in cities. They're exploiter species. We have other species that are probably adapted. So, if you think of a northern flicker, they can do equally well in the aspen parkland or wherever they find a tree. But we do find a lot of flickers in cities and, doing and making a living there quite well as well. But there's some species that we may never ever see in a city. So my guess is the likelihood of us seeing a Sprague's pipit in Regina is probably pretty low. So where urban wildlife research comes in is to really help us understand how to avoid or reduce negative wildlife conflicts and what aspects of a city may be beneficial for wildlife and serve as important management or conservation areas. Now, like I said, a lot of the research generally finds that biodiversity decreases as you go from the outskirts of the city to the interior, but nothing has really been done in sort of the context that Regina finds itself in. Regina is sort of this tree oasis on the prairie. And a lot of those trees have been planted. We have Wascana Park, which has a lot of wetland habitat. So are we going to see those same patterns revealed in Regina? Well, we're not quite sure, and that's why we wanted to start this research project. So this project, we're going to have 15 biodiversity monitoring stations, at least to start, placed along an urban to rural gradient in and outside the city of Regina. So on the map on the right, those red areas are sort of our rural sites. Anywhere from Condi to White Butte to some of the golf courses outside the city. Then we have some areas in yellow that are kind of right on the edge. So sort of a 50-50 urban to rural development gradient. And then we have some sites right in the core of Regina. So all the stations will be placed within city green space. So things like golf courses, parks, cemeteries, natural areas. But we're also going to have some sites outside of Regina's to compare to. The stations are going to consist of trail cameras to take pictures of larger birds and mammals. We're also going to have automated microphones that are going to record bird songs and vocalizations, amphibian vocalizations. Maybe we'll even hear some coyotes howling. And then we're also going to have bat detectors, which record the echolocation calls of bats. And then we can go back and identify what species are here. So the stations are going to be active 4 months out of the year. We just put up a subset of the stations on Friday. So they'll be up for 4 weeks, and then we'll take them down and take all the memory cards with the pictures and the songs on it back to the lab and start to identify what we've seen and heard on the cameras and microphones. And what are we asking here? Well, we're asking some interesting questions about how the urban landscape can promote biodiversity. Does it depend on the amount of green space? How big those patches of green space are in the city? Does the age of the neighborhood matter? How much tree cover is there? Does that matter in terms of influencing biodiversity? On more of an interest level, we're just kind of interested in what species are present in Regina. There's been a lot of surveys in the city here, but can we add to that with taking recordings at night when people aren't typically out doing surveys for this species? We're also interested in what their numbers might be in the city. So, for example, can we get a handle on how many rabbits there actually are in the city? What about cats or free roaming cats? And we can answer some neat questions about how many yellow warblers there might be breeding here in Regina. Because we're going to do it across various seasons, we're also interested in whether or not wildlife use of the city changes from fall to winter to spring to summer. Now, after we're done dealing with the collections, the outreach and education and research, then we can take a little break and sleep. Or do we? And with that, thanks very much for everyone very much for coming out tonight. It definitely feels like fall is upon us, so really appreciate people coming out to the museum in person and for folks tuning in on the website. My contact information is below. Fran had mentioned my website, and that's the link to it there. So I'll leave that up for a couple seconds, but I can't see the chat. But if anyone has any questions in the chat, I can try to answer them. And if anyone has any questions from the audience there, I'm more than happy to answer those as well. So thanks very much, and I'll stop sharing my screen right now.
In the audience here? We've got Ryan running around here with a horrible light tonight.
Yeah, Ryan, when you look at the assessments you're collecting or getting in, are you taking any photographs of those while you're doing all that data collection? Or so you can actually have a visual image of the assessment?
Yeah, so whoops, getting a little bit of an echo here, but I didn't quite catch the whole question, but it sounded like you asked if we also take pictures of things that come in, and yeah, we're just starting that process to take a limited number of digital pictures of stuff that comes in, especially for things that we turn into a skeleton, just so we can kind of maintain that record of what the thing looked like when it was brought into the museum, because sort of at the end of the day, all we'll have is the skeleton remaining. So I hope that was the question. I heard picture and a few other words, but I hope that's what you were asking. If not, I can try again. There was a question in the chat from Gail. She asked, how long will the Urban Wildlife Project be? So Gail, what we're hoping is that this Urban Wildlife Project becomes a long-term monitoring project here in Regina. So we're hoping that it's going to kind of keep going indefinitely, as long as as long as our equipment stays in good shape and we're able to kind of keep up with all the data that comes in. We're hoping that it's going to last as long as possible. The other interesting thing is that there are a couple other cities in Canada, specifically Calgary, Edmonton, and Saskatoon, that also have urban wildlife projects. And so we're hoping that we can collaborate with them a little bit on some of the data that we're collecting here.
Can you hear us on this end, Ryan?
Yeah, I can hear you now. Yep.
Okay, maybe try now,
All
Fred.
right, can you hear me, Ryan? Ryan, are you there?
Yeah, I can hear you.
Okay, one more question.
How do all the birds not run away when they're in those little capsule things?
Can you repeat that question, Fran?
He's wondering how the birds don't run away while they're in the capsule thing.
So how the birds don't run away.
Rot, Ryan, it's how the birds don't rot when they're in the
Oh,
capsules. Yes.
how the birds don't rot. Good question. Yeah, that's it's really an art form to make those study skins and make those skeletons so that they don't rot. So when we prepare them, we try to take out as much of the stuff that could rot as possible. So we try to get all the muscles and all of that type of stuff out and scraped off of the bird so that it doesn't rot. We use some things that will sop up some of the liquids so that doesn't rot. And for the most part, that does a pretty good job at preventing any of our specimens from rotting. The other neat thing is, if you remember that picture that I showed you with all of the cabinets, that room is sealed off from the rest of that building. And that's to prevent bugs from getting in, which can do some real damage to the collection. It also keeps it nice and dark because we know that light can fade feathers and fade fur and things like that. And it's also temperature and humidity controlled. So we try to keep stuff as safe as possible from things that can ruin it. So yeah, rotting though is a real potential problem, and we do our best to make sure that there's actually nothing in the bird to rot when it's in our collection.
Is there anyone else in the audience? I think that's it for questions here, Ron. I'll just get back on the stage.
We have no chats, questions? Or we do?
Nope, just the one from Gail that he answered.
Okay. Well, I think this closes the meeting then. Ryan, that was a fabulous presentation. I thank you very much for all the information.
Thanks very much.
Thank you very much. And thank you, Kirsten, for all your help with the visual.
And Darylyn.
And Darylyn, thank you.